Good morning or good afternoon, everyone, depending on where you are joining us from, and welcome to today's webinar. My name is Rachel Starbuck from the Business Review, and I will be your host. It is our pleasure to have LCS Lohmann Therapy Systems with us today, who will be discussing analytic considerations for testing transdermal delivery systems. Today's guest speakers are Dr. Michael Komenda, who is the Corporate Senior Director and Head of Analytical Development in R&D. And we also have Dr. Iris Schnitzler, who is in Marketing and Sales Intelligence. I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you disconnect, please click on the link that you received via email to rejoin. You may move and resize the windows on your screen to customize your view. All of the icons along the bottom of your screen are the interactive widgets that we have on offer today, so please interact with them all throughout the session. In order to ask questions, you can send them in via the Q&A widget at the bottom of your screen, or please use the top left-hand box. We will allocate around 10 to 15 minutes at the end of a session to address any questions or thoughts that you may have. Please use the help widget if you experience any technical difficulties or require any more assistance. So without further ado, please allow me to welcome Iris.
Thank you, Rachel. Hello and welcome everyone for today's webinar. I would like to start this presentation with a short introduction of our company. Founded in 1984, LTS has become the global market leader for the manufacture of transdermal systems. LTS owns more than 300 patent families, resulting in about 3,000 individual patents. In 2014, we manufactured more than 25 million systems at our two sites. Our headquarters is located in Andernach, Germany, and our US facility in West Caldwell, New Jersey. Today, LTS employs around 1,200 people, and last year we generated sales of 310 million euros. LTS is currently active with 30 cooperation projects. LTS offers transdermal patches for a number of indications, including Alzheimer's and Parkinson's disease, pain treatment, smoking cessation, acute migraine, and hormone products. LTS is a full-service business-to-business partner for the pharmaceutical industry, offering all services from feasibility studies to commercial manufacture for the global supply. And now I would like to hand over to my colleague Michael for the scientific part of this presentation.
Thank you, Iris. Good morning and good afternoon, everybody. What I would like to do in the next 45 minutes is to bring closer to you transdermal delivery systems and their analytical testing. And by the way, I will use the terms TDS or transdermal delivery systems or patches synonymously. So what I will start with is with showing you a specification for a patch and the criteria that you will most likely find in a specification. And I have clustered the methods to control those parameters into methods which are identical compared to testing of other dosage forms, into methods which are somewhat modified compared to what you know for oral dosage testing, and into methods which are totally unique for patches. And in the download version of this webinar, you will find further information in the appendix of this document. So one of the reasons to make this webinar is actually to bring patches closer to a broader audience. So despite the fact that there is an increasing number of compounds and products entering the market, TDS are still a niche product. There is something like 20 compounds which are currently available in the form of patches. And this inherently means that a relatively small number of companies and people have really hands-on experience in the development, manufacture, or testing of analytical patches. So I hope I can provide some insight into analytical testing based on our know-how and experience, as well as based on the recent guideline documents, Most importantly, the new EMA guideline on the quality of transdermal patches, which became effective in June of this year, and also based on USP Chapter 3, which is currently in revision and open for public commentation until end of November. But before we get started, we actually like to get to know better our audience, and we have prepared a short survey and a quick handover back to Rachel.
Okay, thank you very much, Michael. So do you or does your company have any experience in analytical testing of TDS? And if yes, are the tests conducted in-house or by an external laboratory? And the answers are from yes internally, yes externally, or no. So if you could please select the answer that is relevant to you and then click on the submit button. We will give you just a few more seconds to do so. Okay then, so if we just move on to the results. Okay, so Michael, can you comment on these results for me?
Oh, it's interesting. It's a fairly split result, almost evenly distributed. Interesting to see that so many people do have experience, either internally or externally. But I see that we also have more than 40% of people who are not experienced. I think it's too late right now to rework my slides, but I hope those people who are not familiar in working with patches will find it interesting. They will hopefully learn it's not rocket science what we are doing here. And I hope that those who already are experienced will at least find one or two aspects which they maybe haven't heard before or which they will find interesting and which could also trigger scientific discussion after our webinar. So to set the stage, what I will talk about, the first slide shows the most simple form of a patch. It shows a monolithic matrix, a TTS, which basically consists of a backing layer to which a drug-containing adhesive matrix is attached. And on the opposite side, we have the release liner. That is the part of the patch which is removed by the patient prior to the patient applying the patch to the skin. This is what I'm going to talk about, analytical methods which are designed to test those type of patches. What will not be in the scope of this webinar are reservoir-type patches, as well as patches with an active delivery of the drug substance. So for the form of passive patches I've shown, here is an overview, which is non-exhaustive, a list of criteria which you may find in the specification of such patches. And the left column makes reference to criteria which will make use of methods which are nearly identical to other dosage forms, things like appearance or dimensions or even residual solvents. The test applied to patches is almost identical to the control of tablets or capsules. I will not go into any detail for those methods. The middle column shows methods which are somewhat modified compared to what you know for testing other dosage forms. And I will show you some practical examples where the modification takes place. And then we have the third column. Those are criteria which are controlled by methods which are more or less unique for patch testing. And I will go into a little bit more into detail for those methods. So to start off with, HPLC testing is applied for testing patches for various things. It's assay, constant uniformity, chromatographic purity testing, or functional excipients. Basically, the HPLC part of the analytics is identical to what you know of other dosage forms. The key part where patches differentiate from other dosage forms is actually sample preparation. And I will show you on the next slides where practical problems can arise. So when you would like to perform an assay or content test from a patch, the first important thing is to get the drug substance, the API, into solution. And basically, you are left with two general possibilities. So number one, you could extract the API from the patch, or number two, you could dissolve the entire matrix. If you choose to do the first one, which is the most common type of sample preparation, you perform the extraction of the API from the matrix. You need to thoroughly develop and validate certain parameters around the extraction, which include the extraction time or the temperature. It's whether you have to use elevated temperature or not, whether you need to stir or shake your sample, or whether you actually also need to use sonication to have a quantitative extraction from the drug substance from the patch into the solution. And what is important here is, especially during method development, that you do not only use fresh samples, but that you also use aged samples coming from your first stability samples, because what is likely to happen, or what can happen during patch aging, is that you may need longer extraction times for aged patches as compared to fresh patches. This is nothing which is really too uncommon. So this is really subject to your method development. If you choose to not extract, but to dissolve the entire matrix, you're still left with two possibilities. You could either inject the solution as is onto your HPLC, or you can precipitate out the adhesive. If you choose to inject the solution as is, you need to keep in mind that the adhesive is still in solution, but in most cases the adhesive is not so good for your HPLC column, so you need to ensure that you have a good stand time of your HPLC column against the adhesive, which is not a problem in case you choose to precipitate out the adhesive. You can do so by adding an anti-solvent, which keeps the API into solution, but which is an anti-solvent for your adhesive, and it will precipitate out the adhesive, and you can centrifuge then your sample and inject the supernatant, which still contains the drug substance, but no longer the adhesive. If you do this way, of course, you have to thoroughly develop and validate this sample preparation step as well, because you need to ensure that you have quantitatively left the API back in the solution. Some further very practical aspects to consider in sample preparation is the geometry of patches, which can sometimes lead to difficulties. A patch is a flat item which has maybe a large surface, but is very thin. And unfortunately, you need to use a little more solvent for sample preparation than you actually like to use. And in combination with the fact that patches typically only contain a small amount of drug substance, this can make your quantitation limit sometimes a little bit more challenging. There are some other very simple things that you need to consider when working up patches and when doing sample preparation. First of all, the release liner is removed prior to sample preparation, and you need to ensure that the patch does not stick to the flask. You want to stick it to the patient later on, but you don't want to stick it to the flask during sample preparation. Also, for those very inexperienced, train your technicians to not fold the patch sticky side in. Of course, this way the patch will not stick to the flask, but you will not have an extraction in the second place. Also important for assay preparation, when you work up multiple patches in the same flask, ensure that the patches do not stick to each other in this step of the sample preparation. Or you can also choose to do something completely different for the assay testing. That would be to calculate your assay result from content uniformity testing. This is also occasionally applied. Of course, this facilitates the sample preparation step because you will always only have one patch in the flask. However, this is easy for the release testing, but during stability testing, you are left with multiple sample preparations compared to working up simultaneously 10 patches in one flask, for example. Something else to consider, especially important for the impurity testing, is that when you perform the extraction, what you put into the flask is the matrix containing the API as well as the backing layer. This is inherently put into the flask. And typically the backing layer is not a transparent foil, but it will be imprinted with a color. And you must make sure that the color does not interfere with your analyte, which appears logical, But sometimes you need to push your marketing to make an early decision because they could come up quite late with funny ideas of how the patch should look like and which color they would need. So you need to push them to make an early decision, or otherwise you will have to have a very late method development because you have some interferences. Same applies even if you have chosen a color for the backing layer. Make sure you have a proper change control system in place, that you are informed when there is any change in early color ingredient, because you should test whether this interference still occurs or not. The alternative to solving that problem from the beginning would be to swap all the patches and actually to remove the color from the patch prior to sample preparation. Moving on from the test itself to the specification for assay. What you know for specification setting for oral dosage forms, for example, for assay, is that the drug content is specified in a range of something like 95.0% to 105.0%. You can do the very same for patches. That's not a problem. But you can follow also an alternative approach, which I would like to bring closer to you. Unlike a tablet, a patch is not completely consumed during the application time. In fact, there is always a residual drug amount in the worn patches, and this is really something which agencies have an eye on. It is important because also agencies will push you to have this content as low as possible. But the key part is the absolute amount of drug in a patch is not as relevant to the patient as is for a tablet. And therefore, an alternative approach to assay specification is also to specify the drug concentration in the matrix and the dimensions, because this is actually more important for a patch. And typically, the X and Y dimensions, which would be the surface area of a patch, those are important for the dose. And you can see so because the matrix-type patches are manufactured from the same laminate, and those are punched out in different sizes from the same laminate. And the Z dimension, being the thickness of a patch, is typically something which is proportional to the application time. And you can alternatively think of specifying the assay by the dimension and with the concentration. It is not mentioned in any of the guideline documents, but there are products on the market which follow this type of assay specification. It has not been accepted in Japan so far, but it has been accepted to European and US market. So moving on from the assay test to the dissolution or drug release test, again, this is fairly similar to what you know for oral dosage forms testing. There are various types of instruments available, but the most two common ones are the pedal over disc or rotating cylinder assembly. Those are well described in USP chapters 711 and 724. But you must know that there is a certain size limitation in the maximum size of a patch which is applicable to this type of test. The rotating cylinder with the extension set can take up patches with a size of up to 200 square centimeters. So this is not a problem during the patch testing. If possible, you should avoid cutting the patches for this test, and it's the expectation that you test the patch in its entirety. Sometimes larger sized patches make it unavoidable to cut the patch, but keep in mind you can only cut proportional matrix type patches and never reservoir type patches. Frankly, I have not seen a reservoir type patch which exceeds a size of 200 square centimeters. If you would run into that position, you really have to think how to conduct the drug release test for such a type of test. Whereas things appear to be fairly clear for the US, things tend to be a bit more complicated in Europe. The European Pharmacopoeia also describes the dissolution test for patches in a dedicated chapter, that is chapter 294. But you need to be careful. Unlike chapter 293 for testing of oral dosage forms, This chapter 294 does not contain a level testing, and unfortunately it also does not make any reference to chapter 293 as is done in the USP. The USP does cross-reference the two chapters, has not been the case in the European Pharmacopoeia, which didn't bother us in the past. Unfortunately, to make things even more confusing, there is a quote from the new EMA guideline which reads out as the number of samples used to characterize the dissolution profiles should normally be a minimum of 12 units per batch. For routine release, a minimum of 6 units would be accepted. This raised a lot of concern, and I was attending a USP seminar in September in Washington, and there was a colleague from the EMA, there was a representative who kind of clarified this in a verbal statement that it's not the intention to have it always running 12 units, but unfortunately this is only available as a verbal comment, and that's really nothing we have in writing so far. So take care that this could lead to maybe somewhat confusing discussion. On the other hand, very good what has been put into the guideline in relation to other topics, which have been harmonized also with the guideline for the oral extended dosage forms. Likewise, you have to have at least 3 sampling points for the drug dissolution test. The first time point should be set at a time at which around 20 to 30% of the drug has been released in order to exclude dose dumping. The second point should be after a time when 50% of the drug has been released, and the last time point should be set at a point where about 85% or more has been released, which can be quite challenging for some of the products which tend to not release their API so fast. What is also good, at least in the final version of the EMA guideline, is the normally permitted specification range, which is now clarified as being around plus minus 10% of the label content of the drug substance, meaning if the value is set to 50%, the range would be between 40 and 60%. In the original draft, if those of you who were concerned when reading the original draft, it read 10% relative to the accumulated amount released as a given time point, which would have meant if 50% is the midpoint, the range would have been 45 to 55, but this has been revised in the final version to what you can read on the slide here. Some further practical experience for this test. You need to know and also make sure in all discussions with everybody involved that the in vitro release characteristics are completely different to what is happening onto the skin of a patient. There is strictly no in vivo, in vitro correlation for this test. And this point really needs to be stressed and emphasized because this also leads to the fact that the in vitro dissolution time does not need to be as long as the intended clinical application period. So if you develop a 1-day patch or a 3-day patch, the dissolution test does not need to run as long as 1 day or 3 days. I know that in the past, people may have received such requests from authorities, but I encourage everybody to really enter a scientific discussion when coming to this request, because there is no scientific rationale as to why the test should run as long. Sometimes it may need to be as long, but there is no necessity. Our practical experience in terms of the confusion, how many units do I need to pedal, is really our experience is that when you perform the dissolution test with 6 units and applying the level testing as referenced in USP Chapter 724, this is meaningful and has been accepted by the agency so far. So level 1 testing, 6 units, and if you do need to conduct level 2 or level 3 testing, this should be possible to do so. So I'd like to move on to those test methods which are more or less unique for testing. Starting off with a crystallization issue. The picture shown here is publicly available on the web page described below the picture. It kind of pains me to have to show this picture because I was somewhat involved in the development of that product. But crystallization is an issue and can be problematic for patches, and it's clearly an undesired effect which has led to product recalls and also to prescribing restrictions. If you do look into the guidelines, and we will do so more details in the next slide, crystal formation is not included per se. However, the hurdles to having patches or products accepted, which do show crystallization, are extremely high, and this needs to be thoroughly investigated and controlled by suitable specifications. So the new EMA guideline sets out the stage for what the expectation of European agencies are in terms of crystallization. And here are some extracts from the recent EMA guideline, which became effective in June. So clearly, crystal formation is not regarded as a cosmetic effect. It's named as a quality deficiency, which is thought of as likely to adversely influence the in vivo performance of the patch. I'm not always conforming to the likely, but it's at least possible. It's the clear expectation that at point of release testing, a transdermal patch should show no signs of crystallization, unless, of course, you have a suspension patch where the drug is dispersed in solid form. Those patches will always have crystals in the matrix because that is the built-in design. However, if you have the drug dissolved in the matrix, it's the expectation at time of release, no crystals. The guideline concedes that sometimes during shelf life, it is unavoidable that crystallization occurs and that crystals do show up. However, it is expected that you have a satisfactory description and explanation, which you must include in the summary of product characteristics and the package leaflet. And clearly, the shelf life specification for the crystals needs to be fully justified by relevant in vitro drug release and dissolution data, as well as skin permeation data, and as necessary also clinical studies. For those of you who may have had this request, performing a clinical study with patches showing crystallization is far from being an easy task because crystallization is often a somewhat erratic process, and crystals are never really homogeneously across different patches, even from the same batch. So it is really, really difficult to conduct such kind of clinical study with patches containing crystals at a certain and specified amount in comparison to patches without any crystallization. So even if you do want to take the money and time to conduct clinical studies, this is extremely difficult to perform. To me, it sounds more than a theoretical possibility than a practical one. And last but not least, agencies are starting to expect more than a simple visual count and control of patches for crystallization. It is expected that for better quantification, microscopic or photometric methods are preferred. So really, to wrap up things in terms of crystallization, my very personal advice would be avoid them. Avoid them if you can, because from personal and practical experience, I need to concede that it is really extremely difficult to convince everybody that the amount of crystallizations that you may see on your product really does not have any effect on the therapeutic efficacy, even despite you thinking you have sufficient data. This is clearly a critical path for any product with an unclear submission and an unclear feedback from regulatory authorities when you do have a product containing crystals which show up during shelf life. Since we're talking about patches today, one of the most important things, of course, is to specify and measure the adhesive properties. This is one of the key challenges in product development, to have a product which really has good adhesive properties. There are 3 tests which are referenced in USP Chapter 3, which is the peel adhesion test, the release line appeal test, and the tag test. I will show you more details on these tests on the next slides, as well as some other further tests. And there's also a nice literature reference by Paola Minghetti and colleagues. This is a nice comprehensive overview about measuring adhesive performance. If you haven't read this literature, I highly recommend you to read it. What I really do like in USP Chapter 3 is the expectation towards the performance of a patch and what we actually expect from a patch and the adhesive in a patch. This is a really good and condensed expectation on those properties. There is just one missing. What we expect from the adhesives in a patch is that the patient should be easily able to remove the release liner from the patch. That is the prerequisite so that the patient can actually apply the patch to the skin. And when the patient does so, the patch must adhere properly to the skin, and the patch should stay there over the entire period of use, which could be as short as a day, or maybe half a week, or a full week. And after the application period, the patient should be able to remove the patch from the skin without any problems and without leaving any residue on the skin or causing damage to the skin. The only smaller part missing here in this summary is that the patient should be able to remove the patch from the pouch without any problems, but it's mentioned somewhere else in terms of code flow, and I will come back to that a little bit later. So how do we perform the measurements of all these physical tests? How do we all control these physical parameters? For most of the tests, reference is made to norms of the American Society for Testing and Materials, to the ASTM norms. However, you need to be very, very cautious because those norms were not designed for testing patches. Those norms were designed for testing adhesive tapes. And I will show you in the next slide where the problems arise when testing something as small as a patch as compared as to testing larger tape strips. What's also important to mention is that those ASTM norms, they reference a norm climate, which is 23 degrees Celsius and 50% humidity. So it's the expectation that you conduct the measurement and have the sample preparation and conditioning under those norm climates. And the picture shown here shows how we have set it up in our labs. It's actually a room inside a larger room, and this room is not only controlled for temperature but also for humidity, which can be sometimes a little bit more complicated to set up. So starting off with maybe one of the most important tests, which would be the peel adhesion test. The peel adhesion is specified as the force which is required to remove the patch after it has been attached to a standard substrate surface. In our case, you see it's a stainless steel plate. There are also some other materials mentioned in the ASTM norm. You see on the picture that the patch is not directly attached to the instrument, but there is a green supportive tape. So the supportive tape is attached to the patch on the one side and to the arm of the instrument on the other side. And in the test shown on the picture, the test is conducted at a 90 degrees angle. The ASTM norms also reference a 180 degree angle, and it's really up to you to decide which test is most appropriate for your specific product to conduct. So what makes this test challenging? First thing, very simple, patches can be fairly small. The patch shown on the picture was, 20 or 30 square centimeter patch, but patches can be as small as maybe 2 square centimeters or even smaller. And this makes this test very challenging. And you also need to specify that you typically discard the first few and last few millimeters of your measurement run, and that you average over the mid part of your measurement, and you need to thoroughly specify this in your method so that the analyst cannot choose over which range to average. This must be clearly specified in your analytical method. And there has been a recent finding expressed by the FDA in relation to that method. When conducting the test according to the ASTM norm, there are plates commercially available which come certified as fulfilling the ASTM norm. But you will find that when testing various plates, you will find difference in the results. And this is, of course, subject to your method development and simply knowing your overall method variability. You need to really conclude this as part of method development. What is the variation I will see when I try to keep everything as constant as possible? As mentioned already earlier, you cannot only use stainless steel, you can also use other materials, for example, aluminium. However, at least we have not found any supplier which certifies aluminum plates as being certified conforming to the ASTM norm. So you do need to install some kind of qualification strategy for other material plates. What's also important, those plates are not single-use. You will use them frequently. Of course, you will have to clean them in between the measurements and ensure that they are free of any residues. Actually, you must also check each plate after the measurement and check whether you do have adhesive residues on the plate after the measurement, because if you do have, you probably have not measured peel adhesion, but you probably have a cohesive failure. And this is important to differentiate because you want to measure peel adhesion, but maybe not necessarily a cohesive failure. For those who have not conducted the test at all, you must know this test has a much higher variability than you experience in the chemical tests. So this also means you will need to set your specification properly. Always only specify the mean results, not individual results. Those can vary significantly and much more than what you know from chemical tests. Second test, the release liner peel test. Here you basically measure the force which is required to remove the release liner from the adhesive part of the patch. That is the first step that the patient does after the patient has removed the patch from the pouch. And the patient must be able to remove the release liner from the patch prior to application. There is no dedicated ASTM norm available for this test, and I also didn't include any picture here in the webinar. But the basic principle is the same as for the peel adhesion test I've shown before. Also some practical considerations. You can run both tests, the peel adhesion test and the release liner test, on the same instrument. However, you need to ensure that the instrument is qualified over the entire range of the results that you will see. Why do I mention this? Typically, the peel adhesion results will be on the order of few or several newtons. However, the release liner peel test results are often only a few centinewton, and you need to make sure that the instrument is qualified over the entire range of the results that you will see. The third bullet point I've already mentioned for the peel adhesion test. The last two bullet points are actually not only addressing analytical colleagues dialed into the webinar, but also to drug product development colleagues. It's important to make a good selection of the release liner, and it needs to be well compatible with the adhesive. This is something very important because there can be a tendency upon aging and storage of the patches that a release liner can block with the adhesive part of the patch, and that is something important which need to be tracked thoroughly during stability testing. And what can also happen is in the instrument, which has a constant movement, constant speed, is that you do not necessarily see the blocking as compared to a manual removal of the release liner. So it is important that analytical colleagues do make their development colleagues aware in case they observe blocking in any kind of sample preparation. Moving on to the next method, that's the tech test. Various types for tech tests are referenced in USP Chapter 3. One of them is the rolling ball method. Tech would be the property of an adhesive to form a bond with a surface upon only brief contact and under light pressure. With the peel test, you apply a certain pressure. With the tech test, you apply very little or almost no pressure at all. And one of the tests which is referenced in the guideline documents is the rolling ball method. And here we brought some videos. You already see the picture. The rolling ball means that you have a ramp, which you see on the left side. The ball will come from the left, and at the bottom of the ramp, you see a round-shaped patch. And right next to it, there is a ruler, because the intention of this test is to measure the distance traveled. So maybe if we can run the first video. Okay. Takes a few seconds to load. Ball is coming from the left, rolls right over the patch. Maybe we can show this again. It's very fast. Again, ball is coming from the left, but it rolls right over the patch. Okay. Also, the opposite can happen. That is shown on the next video. Again, ramp to the left, ruler on the right. Ball is coming from the left. And you will see the ball will roll down the ramp, and it stops immediately. In either case, it's really not meaningful to measure the distance traveled. This is really kind of hard to measure. In this case, we never have seen, or very rarely seen, this test work in a meaningful way for a patch. But there are other methods available. For example, the probe tag tester, which is shown here. Probe tag measures the force which is required to separate a stainless steel probe from the adhesive layer of a patch. There are various instruments available. Some instruments have the probe coming from the top. In our case here, also very small video, you will see that there is a static probe faced upwards, and the patch is moved downwards, has a contact to the probe for about 1 second, is moved upwards again, and the tag is measured as the force which is required to remove the tip of the probe from the patch. Again, important to mention, this method is not referenced in any specification of our marketed products, but we are using this test so far exclusively in an R&D environment. Further tests for physical tests include static shear adhesion, which is referenced also in the new EMA guideline. Static shear would mean that you have a constant stress to the patch or a part of the patch. The constant stress is applied by putting a weight, a constant weight to the patch. And what you measure actually is the time which is required for the weight to pull down the patch from a more or less vertical panel. Practical experience with this test, results can vary very significantly, even more than for the peel adhesion test. And actually, if you have a closer look into the ASTM norms, even the authors of this norm must have known it, because they do not require you to calculate the arithmetic mean, but they ask you to calculate the mean of the logarithmic values, which already tells you a little bit about what their expectation is towards the variability of this method. And basically, this buffers extreme values, especially the higher values. So this is truly important. Only specify the mean result and set the specification with care. And some advice for specification setting. You will find that the test results will depend on the quality of your adhesive that you use. And typically you will have also for your starting material for the adhesive a certain specification range, and make sure that when setting the specification for this test that you have had access to adhesives covering the entire range of the specification, because this is really important when setting up the specification. Something else to mention, for the very simple monolithic patches, the test can be interpreted. However, if you do have a patch which is manufactured as a multi-layer system, this test is very difficult to interpret, because what you will see will not necessarily be the cohesion of the matrix, but it could be the shear between the different layers of your product. So it is really difficult to interpret from multi-layer systems. Something else which you must keep an eye on is the so-called code flow. Code flow refers to the dimensional changes or deformations of your patch matrix beyond the boundaries of the patch, which is especially important during stability testing and during aging and storage of your patch. Code flow happens when you have an excess of adhesive forces over cohesive forces, meaning that when you have a patch matrix which is too soft, which is good for your adhesion on the skin, a too soft matrix could mean that during storage it will flow out and ooze beyond the boundaries of your patch, and this can make it difficult for the patient to actually remove the patch from the pouch. So you need to control your adhesive cohesive forces, and code flow will show you where you are in this sector. For the analytical testing of code flow, there is no golden rule. There is no really gold standard that I would recommend. There are various methods available, all of which have their pros and cons. Several methods have been accepted by agencies so far. A very simple quantification method would be the so-called distance method, which measures the distance of the code flow traveled at predefined and evenly spaced positions of a patch. Of course, if you have a round-shaped patch, make sure to specify the orientation in your method. A little bit more sophisticated is the so-called overall area method. In this case, you would take an image of your patch, and you would use an image analysis software to quantify the amount of code flow which has traveled beyond the boundaries of the patch. Third alternative, you can use a kind of swap method by stripping the code flow part of the matrix. Alternative to that, you can even die cut and punch out the original size of the patch, remaining the code flow outside of your die cut and quantifying that. Or a very simple but very effective method is also to have a reference plate which is somewhat larger than your patch itself, but which would specify the maximum accepted area and maximum accepted code flow for your specific patch. Pouch tightness is something you may want to consider, especially important for patches which do contain an API which is subject to oxidation, or which is maybe of particular concern when exposed to water vapor. This can be an important test, so you want your pouch to be tight. Also here, various possibilities, starting with a simple visual detection of pinholes in your packaging. You can test the oxygen content in your pouch, which is possible if you purge, during manufacture, if you purge the air with nitrogen, you can check whether you have any increase in oxygen. Very simple and effective test is to put the patch in an exsicator and apply a vacuum and see whether the patch inflates by the residual amount of air left in the patch, and that it stays inflated inside the vacuum. A test which is a little bit more sophisticated, and it's not only connected to pouch tightness, but mainly to seal integrity, is to insert a colored ingredient, a colored solution into the pouch. That can help you when looking for micro cracks or seal deficiencies. What you may want to do is to, at least during method development, is to make a positive control, to check what is the smallest deficiency in my packaging material that I will be able to detect by my method. You should be able to know this. And this is really a meaningful parameter during method development or during product development, but not necessarily as a permanent quality control method, because from my personal impression, pouch tightness itself is not a quality parameter. You will see the quality parameters that will be influenced by untight pouches if you have an increase in impurities, for example. But pouch tightness itself is not a quality parameter, but it can be supportive in finding a root cause in case you do have problems with other parameters. A test which is currently not specified for patches, but which is extremely important in an R&D environment during patch development, is the skin permeation test. For those who are somewhat familiar with the patch testing, performing Franz cell type experiments with skin permeation is the most powerful tool during development to see if you have a compound which can permeate through the skin. This is not a quality control instrument, it's an R&D instrument. However, I've written here not yet quality control because we have to concede that the recent EMA guideline puts a lot of emphasis on this test, and actually there is a complete appendix dedicated in the guideline to this test. And although the guideline itself concedes it's not a quality control tool, there was also a sentence in there which reads like, in vitro skin permeation should be consistent throughout the shelf life of the drug product to provide supportive stability data of product performance on storage. And there is kind of a tendency of agencies to rely on data, so probably you will be asked for data for skin permeation results during stability testing, and agencies could actually reference this guideline in the future. So coming to an end of the presentation, to wrap things up, I hope I was able to show you that it's not rocket science. There are meaningful tests available to control the quality of patches. Most of the tests are familiar to you. Some tests are modified. There are other tests which are unique. Some tests still require further optimization. Some tests do work in an R&D environment, have not yet found their way into quality control for market supply, but that may change as we move on. And as a short outlook, it would be interesting to see when more competition and more companies enter the market and enter the field of patch development and manufacture, that there will be further and continuous improvement of those methods. And we are involved in discussions with agencies, EMA, USP, and pharmacopoeia discussion concerning patch analytics to really further improve those methods. And we are bringing in our knowledge and expertise in order to also protect our customer interests. I think I've extended the time a little bit. I hope you found it interesting. This is basically the end of the presentation part, and I think we still have some time left for a couple of questions. And I'm handing over back to Rachel again.
Okay, thank you very much, Michael. So thank you to everyone who has already sent their questions in. Please continue to send in your questions via the Q&A widget at the bottom of your screen, or please use the top left-hand box. So I know we have had a lot of questions in, so we will try and get through as many of these questions as possible. However, if we don't have time to answer all of them in this webinar, then Michael and Iris will get back to you at a later date. So for your first question, for the assay testing of patches, will the release liner also be extracted?
Good question. The answer is no. For a simple reason. When the patient applies the patch to the skin, the release liner is removed. So it's the clear expectation, whatever quality parameter you're testing, for example, assay, it's the expectation that the patch that the patient applies to the skin contains the amount of assay and drug which is really specified. If you would also extract the release liner, then you would probably confirm that the patch does contain the amount of the labeled drug substance. However, it is important that the part that the patient actually applies to the skin does contain the spot. Okay?
Yes, okay, thank you very much. So for your next question, can some tests be performed on the stage of the laminates rather than testing on individual patches?
Yes, that's possible. For the monolithic matrix type patches, I didn't show how those are manufactured, but basically you manufacture a laminate and you punch out individual patches out of the same laminate. And yes, it is possible, has also been accepted by agencies, to not conduct all tests on the patch, but to conduct some tests on the stage of the laminate. What I would encourage you to do is to make some kind of risk matrix. You should have a look at your manufacturing process and check which part of your manufacturing process can influence which part of your specification. And if you end up with the scientific rationale that it is possible to control things on laminate stage, yes, you can do so. For example, residual solvents. There is no reason why this must be controlled for each patch size, but based on the manufacturing process, it is possible to conduct things like residual solvent tests on the laminate stage. Yes.
Okay, thank you very much, Michael. So we probably have time for just one more question. So we found TAC test results with flat face probe had large variation. Is the large variation normal for TAC test? And if not, is there any recommendation to eliminate the variation?
We see a large variation that depends. Depends on what your expectation is. It can easily be several 10%, and even more, especially when you have low values. As mentioned earlier, the chemical tests always have a much smaller variation than you see in any of the physical tests. But this does not mean that the test is not suitable for the intended purpose. It's really depending on what your expectation is for the specification and how you set up the specification. Yes, it is not uncommon to see a high variation, especially a high relative variation for very small tech results, but that does not mean that the test is not suitable for its purpose.
Okay, thank you very much, Michael. So we probably do have time to squeeze in one more question. So when performing skin permeation experiments as part of stability testing, do you use the same skin specimen throughout the stability study?
Difficult question. Answer is no. You cannot do so because typically the stability study runs for as long as like 2 or 3 years. And you cannot use the same skin strip. Even if you would deep freeze it, it would change its characteristics over time, and skin, even frozen skin, is not stable over such a long amount of time. There are 2 alternatives. You can perform experiments on the same skin strip, but in this case, you cannot use the same batch. You would have to compare fresh patches from batch number 1 and old batches from patch number 2. In this case, of course, you're not having a view on the patch aging of the very same batch of TDS. The other alternative is to take into consideration the biological variation which you see from skin strip to skin strip, and consider this variation up front, and then conduct the experiment on different skin strips. But basically, it will not be possible to say, I run the stability test on a single batch of patches and use the same single skin strip throughout the 2 or 3 years of the stability study. That will not be possible and yield good scientific results.
Okay. So thank you very much, Michael. So sadly, that is all we have time for in today's session. So if we didn't get round to answering your question, then Michael and Iris will get back to you at a later date. So that just leaves me to thank Michael and Iris for what was a great presentation, and thanks to LTS Lohmann Therapy Systems for sponsoring this session. To the attendees, you will receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access this through our website, which is www.business-review-webinars.com. So thank you once again to everyone, and I hope you all have a nice day.